Good morning, everyone. It's wonderful to see you all here this morning, I have to say it's thrilling to be able to introduce our distinguished visiting lecturer today and to kick off our year-long series with Dr. Eric Kandel. I get the unenviable task of introducing a man who needs no introduction. I'll try to keep it brief, so we leave plenty of time for the talk. As I'm sure all of you know, Dr. Kandel is at Columbia University, where he's university professor. He's a Copley professor and director of the Copley Institute for Brain Science, co-director of the Mortimer Zuckerman Mind, Brain, Behavior Institute, and an investigator at Howard Hughes Institute. Dr. Kandel graduated from Harvard and then was off to NYU School of Medicine, trained at NIH, and then was back to Harvard. He joined the faculty at Columbia in 1974, where he was the founding director of the Center for Neurobiology and Behavior. So I'm sure, as everyone in the room knows, Dr. Kandel's research has been concerned with the molecular mechanisms of memory storage, first in aplasia and now in aplasia and mice. More recently, he's expanded his work to touch on a number of topics that are near and dear to our hearts here in the Department of Psychiatry, including PTSD and addiction, nicotine, alcohol, marijuana, and cocaine addiction. So his research clearly has had a profound impact on our field, on all of neuroscience. His work has really helped shape the field of neuroscience and of psychiatric research over 4 decades. The associated list of accolades is pretty unbelievable, but in addition to 23 honorary degrees, membership in the National Academy of Sciences, a foreign member of the Royal Society of London, member of the National Academies in Austria, France, Germany, and Greece. He's been recognized with the Lasker Award, with the Heineken Award, with the Harvey Prize, the Wolf Prize, the National Medal of Science, and of course with the Nobel Prize for Physiology or Medicine in the year 2000. Exactly. Yeah, can we... I think what has always amazed me about Eric, there are many things that have always amazed me about Eric, but one of them has been the impact that he's had outside of the laboratory in addition to the remarkable work in the laboratory. So he organized the neuroscience curriculum at Columbia. He's the editor of Principles in Neuroscience, which is the standard textbook in the field now in its 5th, soon to be 6th edition if we all get our chapters in. Exactly. I'm talking to you. All right. So his books for the general public have been equally remarkable. In Search of Memory, The Emergence of a New Science of Mind, which won both the LA Times and the U.S. National Academy of Science Awards for Best Book in Science and Technology, 2008. In 2012, he wrote The Age of Insight, The Quest to Understand the Unconscious in Art, Mind, and Brain, from Vienna 1900 to the present, always taking on small and manageable topics. This won the Kresge Prize in Literature, which is Austria's highest award, literary award. He has a new book, Reductionism in Art and Brain Science, Bridging the Two Cultures, published by Columbia University Press and just out. So I'll just say briefly that I think, again, it goes without saying that Dr. Kandel is really the apotheosis of the physician-scientist in psychiatry. I think he has been the most important role model now for generations of investigators seeking to understand the basic mechanisms of brain function and psychiatric illness. I certainly count myself in that group. It's difficult really to quantify how important it's been for psychiatry over these years to have him as a symbol of the level and quality of research that is possible in our field. It is really, we're so grateful that you agreed to come and kick off our series this year. Thank you, Eric. Dr. Eric Kandel.
Fantastic. Thank you, Mike. Thank you, Matt, for that marvelous introduction. I just wish my parents were alive to hear it. My father would be proud and my mother would have believed it. How many of you are in the Department of Psychiatry? Please raise your hand. You're extremely fortunate. Matt Stead is without doubt the most powerful chairman of psychiatry in the country. I was asked about his selection here and I was overwhelmingly enthusiastic, as was everyone else who was asked. Matt is one of the few when he got this position, I said, "You're the only chairman of psychiatry who can read and write." Because the level in the field is not as high as it should be considering the problems that we confront. So I was very honored that Matt invited me to come here. So I'd like to sort of tell you a little bit about age-related memory loss, but try to put that in a broader context. Let me give you a little bit of a background for those of you who don't think about neuroscience all day long. The modern study of memory began with the question, can any memory be localized in the brain? It was really Wilder Penfield who realized that the brain does not have any pain receptors, and therefore if you infiltrate the scalp, expose the skull, open up the skull, you can stimulate any area on the surface of the brain without causing pain to the patient. And since he specialized in removing scar tissue from the brain, he had to stimulate surrounding areas to make sure he was not removing something which is critical for function. So in the course of stimulating different areas, he found that you could localize areas that elicited memory storage. And he found that these were in the medial temporal lobe and a structure deep to it called the hippocampus. And this was really confirmed when William Scoville, a neurosurgeon in New Haven, formed a bilateral removal of the medial temporal lobe in the famous patient H.M. in 1953. And he found that he cured him of his seizures, but left him with the most profound memory loss you could possibly imagine. This was studied by Brenda Milner, who characterized his deficit in some detail. And she found that there was a surprising number of things he maintained. So you could remember experiences prior to surgery. Remember going to elementary school, to high school, his job for a while. So long-term memory storage of things that occurred some time ago is not stored in the hippocampus. If you gave him a telephone number, 884-5447, he could repeat it. So short-term memory also did not require the hippocampus. What the hippocampus was required for is to convert new short-term memories to new long-term memories. But what was surprising is that even though there was this memory loss, there were certain kinds of learning that he could do perfectly well as you and me. For example, motor tasks. If you asked him to do the outline of a star, Leonardo da Vinci-like, by not looking at his hand, not looking at the pencil, not looking at the paper, but looking in the mirror, he would make a number of mistakes at the beginning, but get progressively better, so that on Wednesday he was much, much better than he was on Monday and Tuesday. He was as good as you and me in any of these tasks. But when you asked him, how come you're doing so much better on Wednesday than you did on Monday? He said, what are you talking about? I've never done this before in my life. The amazing thing is that the kinds of things that could be remembered could be really quite complicated. So for example, once you learn how to play a musical instrument and you master it, you can have bilateral lesions of the hippocampus and still play it. And Clive Waring is a classic example of this. He has a complete loss of explicit memory.
How many years have I been ill?
About 20.
About 20. Can you imagine what to have one night 20 years long with no dream? That's what it's been like. Just like death. No difference between day and night, no thoughts at all. In that sense, it's been totally painless. Which is not something which is very desirable, really, is it? Because it's precisely like death. If you have no senses of pain, you have no sense of any kind working either. I don't remember sitting down on this chair, for example, or this settee as it is. That was unknown to me. I've never seen a human being since that's the first photograph I've seen of anybody.
And who is that
It's
photograph?
one of my sons. I can't remember his name.
Terrible. But despite this, he plays the piano as well as he ever did.
Clive Waring has one of the worst cases of amnesia in the world.
I know it's like we're dead now. Day and night the same. Right. No difference between dreams and anything like that. No sense at all. The brain has been totally inactive. no dreams, and no thoughts of any kind whatever.
Clive was a renowned conductor living in London when he was struck down by a virus in 1985. Parts of his brain were completely destroyed, including his memory. However, his ability to play music is unaffected.
Even to conduct.
Do you feel different when you play music?
I've never heard a note since I've been ill.
Isn't that amazing?
I always liked to play music. When you're unconscious.
You played us some music about 2 minutes ago.
Not known to me. Totally unknown. I've never heard a note yet.
By the way, there are seats up front, and you wouldn't bother me at all if you made yourself comfortable and sat down. Please. So there are two types of long-term memory, implicit and explicit. Implicit is unconscious recall, playing the piano without even being aware of it after you've mastered it. And explicit is conscious recall, like recalling your first date or whatever. And implicit memory involves the amygdala, the cerebellum, in the simplest cases, the reflex pathways themselves. And explicit memory for facts and events involves the medial temporal lobe and the hippocampus. And what are the molecular underpinnings of that? What are the mechanisms whereby storage occurs with each of these forms? And this actually makes a very interesting general point. You people are too young, almost without any exception in the audience, to realize what a major impact molecular biology made, because a lot of things that turn out to appear very, very different turn out to share molecular mechanisms. So the anatomical system may be different, but the molecules used in these different anatomical systems to mediate a particular process could be quite homologous. So with both implicit and explicit memory storage, there are certain features in common. There are two stages of memory storage. There's a short-term memory, which lasts for minutes, and long-term memory that can last for days, weeks, in some cases the lifetime of an individual. Two, repetition converts short-term to long-term memory. Now, it's possible that a single event can be so powerful that it's carried into long-term memory, but almost invariably, if you repeat something, you're much, much more likely to put into long-term memory. And long-term memory differs from short-term memory in requiring the new protein synthesis. And there are a number of systems that have been used historically for studying implicit and explicit memory storage. Obviously, any animal that shows explicit memory storage also shows implicit memory storage, but there are even simpler animals than the mouse, for example, to study implicit memory storage, and Drosophila and Implicit have been used extensively for these purposes. In Implicit, people have studied learned fear, a form of which is called sensitization, where you just shock the animal's tail, you scare the hell out of it, so almost any reflex that you elicit will be enhanced. Explicit memory can be studied in the mouse, and the rat, and of course in humans. And by and large, what has been studied in the mouse often relates to space and the hippocampus. Let me say something about Implicit. As you can tell at a glance, this animal is extremely beautiful, highly intelligent animal. In addition, it's been highly recognized. This was sent to me before I won the Nobel Prize, and I showed it in my Nobel Prize lecture because there was a general feeling among my friends that the animal deserved it a hell of a lot more than I did. This is your brain and mine, 86 billion neurons. Implicit, by contrast, has 20,000 nerve cells. This is Alan Busbaum's brain, this is Implicit brain. Sometimes they get them reversed, Alan. These 20,000 neurons, many of them are very large, millimeter in diameter, and many of them are uniquely identifiable. So you can give them a name and return to the same cell in every animal of the species. R1 is on the right side of the ganglion, L7 is the left side of the ganglion, etc., etc. So you can deal with identifiable cells that are reliable in their physiological properties and in their position. We studied a very simple reflex, a withdrawal reflex, gill withdrawal reflex. This is a dorsal view of an Implicit. It has a external respiratory organ, the gill, which is covered by a sheet of skin called the mantle shelf, which is there for protection. It covers the gill. It has the residual shell that a plesia has, and that ends in a fleshy spot called the siphon. If you now apply a weak tactile stimulus to the siphon, you get a nice withdrawal of the gill, sort of a simple withdrawal reflex. If you now give a shock to the tail of the animal, so you scare it, and give the same tactile stimulus again, you now get a more powerful withdrawal. So if I bang on this table, those of you who startle will respond more powerfully to even a neutral stimulus than you did previously. One can work out the neural circuit of this. There are 24 sensory neurons that pick up directly from the siphon skin. These sensory neurons make direct connections to the motor neurons, and the motor neurons make direct connections to the gill. When you stimulate the tail, you activate a modulatory system, serotonergic, like the serotonergic neurons in your brain, that acts on the sensory neurons, including on the presynaptic terminal, to strengthen the connections between the sensory neurons and the motor neurons. If you give a single tail shock, you activate the cyclic AMP-dependent protein kinase in the presynaptic terminals of the sensory neurons, and you have a covalent modification that gives you a transient strengthening of connections between the sensory neurons and the motor neurons. If you give a repeated tail shock, you turn on gene expression mediated by Krebs, which gives rise to the growth of new synaptic connections. When I first saw this, I realized that this explains psychotherapy. And since modesty has never been my problem, I wrote an article for the New England Journal called Psychotherapy and the Single Synapse, because I realized that psychotherapy must be a biological treatment. It produces long-term changes, at least produced long-term changes in me. I used to be even more obnoxious before my analysis than I am now. And it really gave me sort of an insight in how these processes are likely to function. And in many cases, this growth of new synaptic connection is mediated by Krebs. But as you know, a number of studies have now shown that you can, for example, with depression, Helen Mayberg's classic studies have shown you can localize with imaging abnormalities in the cingulate cortex with depression. And if you respond to cognitive behavioral therapy, that abnormality is reversed. So you can show that psychotherapy is a biological treatment and it produces a reversal of these anatomical changes. Moreover, the growth that occurs, particularly in implicit, is quite dramatic. This is a sensory neuron and a motor neuron before long-term sensitization training. And this is the growth of synaptic connections as a result of this. So I think that this is really a very important concept in psychiatry to realize that this obviously is occurring to varying degree in psychotherapy, and what we want to understand is, can we use this in a reliable way, and what regions can we use in a reliable way in order to evaluate the outcome of psychotherapy, and also to see in what circumstances we might use additional treatments or alternative treatments to psychotherapy using pharmacotherapy. We were able to work out the molecular mechanisms of in Implicit. Tail stimuli activates serotonergic cells. These activate a receptor that activates an adenylcyclase, increases the level of cyclic AMP, activates the cyclic AMP-dependent protein kinase that enhances transmitter release from the presynaptic terminal. With repeated stimuli, the cyclic AMP-dependent protein kinase translocates into the nucleus, and there it activates Krebs-mediated transcription, and that is a two-step. Not only does it activate Krebs1, but it removes a gene repressor, Krebs2. And that's mediated by MAP kinase. MAP kinase phosphorylates and removes the restraining action of Krebs2, allowing the activation by PK of Krebs1, which activates gene expression, giving rise to the growth of new synaptic connections. There now is parallel information on explicit memory using mouse models for people, places, and objects. This, as you know, involves the medial temporal lobe and the hippocampus. And this is really quite spectacular. I don't know whether this holds true for San Francisco. It doesn't hold true at all in New York. In New York, if you ask a cab driver to take you down Broadway from 100th Street to 110th Street, he said, which direction do I go? In London, you really need training in order to be a cab driver. And if you ask a cab driver, how do you go from Hyde Park to Primrose Hill, his hippocampus lights up. And after he drives the cab for a while, his hippocampus gets larger. And the longer he drives the cab, the larger his hippocampus becomes. And when he retires, his hippocampus shrinks. So what is the take-home lesson from that? Matt? Alan? He and I are in the same way. I shouldn't put you in the boat. I'm twice as old as you, but you're getting there, kid. Don't retire. Hippocampus is also essential for explicit memory for space, which you can study in a variety of ways in the mouse. And this involves, the pathways within the hippocampus, from the entorhinal cortex to the dentate gyrus to the hippocampus. And these become strengthened with learning, and they all show variations of long-term potentiation. And long-term potentiation shares a number of similarities to implicit memory storage. The early process, the short-term process, is different. It involves a different transmitter, glutamate, an NMDA receptor being activated, allowing calcium influx, activating a calcium calmodulin-dependent protein kinase to phosphorylate substrate proteins. But long-term memory involves a modulatory transmitter, either dopamine or serotonin, depending upon the system, that then activates an adenyl cyclase, PKA, CREB1, removing the inhibitory constraint of CREB2 and turning on downstream genes for the growth of new synaptic connections. So there are really some parallels between implicit and explicit memory storage on the molecular level. And this is, you really represent two generations after this, but in the early days what really blew us away was to see these processes that were so different, like implicit and explicit memory, turn out to have a number of really powerful similarities when you began to study them on the molecular level. You could block PK, for example, and show that under those circumstances you would switch off long-term memory. So both implicit and explicit memory storage involve a modulatory transmitter, the activation of CREB, removal of inhibitory constraint of CREB2, and the activation of downstream genes that are important for the growth of new synaptic connections. Let me then turn with this physiological background to look at memory disorders in the aging brain. Now, when I was a medical student, we never studied this topic. Why do you think that's so? Do you know? Guess. People didn't live long enough to have age-related memory loss. You got to live to a long age in order to get it. So memory disorders in the aging brain. It's a relatively recent phenomenon. 1900, the average lifespan was 50 years. Now, the weakest sex, men, 76 years. Strongest sex, women, 81 years. And now you're beginning to see age-related memory loss of various kinds. And you can see that Alzheimer's disease, which we practically never saw as medical students, that's because people had to be in their 70s to get it. There is an early genetic form of early onset of Alzheimer's disease, but that's very, very rare. We knew about that. But spontaneous Alzheimer's disease occurs to people in their 70s, 3%. But in the 90s, half the population has it. It's really an epidemic. But in addition, as people age, we began to see another sort of weakening of memory with age, which had some differences and also some similarities to Alzheimer's disease. And one didn't know, is this an early stage of Alzheimer's disease or is it a separate entity? It was euphemistically called as a separate entity, benign senescent forgetfulness. So one of the issues I've been interested in is, are they distinct entities, or is benign senescent forgetfulness an early phase of Alzheimer's disease? And so my colleagues and I looked at three aspects of this: the age of onset and the progression, the anatomical localization, and the molecular defects. One of the things that really struck me is you could go to mice which never have spontaneous Alzheimer's disease and see whether they have age-related memory loss. And boy, they've got it in spades. So here's a spatial memory task for a mouse. Does extremely well. The mouse lives to be 2 years of age. Does extremely well in the first year of life, but at 12 months it begins, at midlife, begins to show a dramatic decrease in memory, and 18 months, much of its memory is really gone. And one can begin to look at where this is located, and Scott Small, a colleague of mine, did this at Columbia. One knew that Alzheimer's disease originated in the entorhinal cortex. Age-related memory loss has its onset in the dentate gyrus. So aging and Alzheimer's disease have different sites of activity at the beginning. So to study this a little bit better, we turned to gene chips, and using gene chips, we looked at the expression of genes in the dentate gyrus, this area that is involved in age-related memory loss, to see whether there's anything characteristic that changes in the dentate gyrus that doesn't change in other areas of the brain. And we came across a number of transcripts that changed, but the one that really stood out most was a gene called RBAB48. This changed selectively in the dentate gyrus. Here's the level of the messenger RNA and level of the protein, and no change in the entorhinal cortex or any other region of the hippocampus. What is RBAB48? Turns out RBAB48 is part of the switch for converting short-term to long-term memory. The switch begins with PKA activating Krebs, phosphorylating it. Krebs activating the Krebs, the phosphorylated Krebs recruits the Krebs binding protein, and that recruits RBAB48, which leads to the chromatin remodeling. So this is really a significant component of the switch that converts short-term to long-term memory. Humans allow correlations, but to look at causality, you have to go to experimental animals, and we did this in mice. We asked, is the level of RBAB48 also selectively decreased in the dentate gyrus of aged mice? So here we looked in a young mouse, an old mouse, a clear decrease in RBAB48, selective to the dentate gyrus. Other areas of the hippocampus did not show that. We can now manipulate it in the mouse. Here's a young mouse and an aged mouse doing an object recognition task. If you put a mouse in an enclosure and allow it exposure to two identical objects, it'll spend an equal time with each. But if you now remove one of the objects, the familiar ones, and give it a new one, a novel object, it spends much more time with the novel object than with the familiar one. So it has curiosity and novelty. And this is something which requires memory. This is a young mouse, spends a lot more time with the novel object, and old mouse doesn't give a damn about it anymore. Moreover, if you take a young mouse and inhibit RBAB48, it acts like an old mouse. And if you take an old mouse and upregulate RBAB48, it acts like a young mouse. So it can really affect memory in a profound way by altering the expression of single genes. Does inhibition of RBAB48 in mice recapitulate the imaging pattern of the dentate gyrus dysfunction? And Scott Small looked at this. This is the normal dentate gyrus, and this is what happens with RBAB48 inhibit. It looks exactly what you see in mice and also in people, that there is less activity in the dentate gyrus with age. So to summarize, the clearest evidence so far that age-related memory loss and Alzheimer's that is linked are that age of onset, age-related memory loss begins earlier, so many of you in the audience have some degree of it. The anatomic localization is different, dentate gyrus for age-related memory loss, Alzheimer's disease for the entorhinal cortex, and the molecular defect is different. Humans show a linear decrease in RBAB48, messenger RNA protein. Old mice have a decreased RBAB48. Young mice reduced RBAB48 have a memory deficit. Alzheimer's don't show such abnormality. Oh, aren't you nice? I still have some water here. But you're giving me a hint, is that right? Okay. Are there hormonal factors that contribute to the hippocampal dysfunction? with cognitive aging, and might exercise help overcome these blood-borne insufficiencies. And there's very good evidence now that has emerged that suggests that, number one, that bone is an endocrine gland. It releases a hormone called osteocalcin. Osteocalcin acts on many tissues in the body: the pancreas, muscle, the liver, testes, and the brain. In the brain, it gets through the blood-brain barrier, and it has effects on mood as well as on memory. So we asked the question, does osteocalcin affect age-related memory loss? And we found that direct administration of osteocalcin into the dentate gyrus enhances contextual fetal discrimination both in young animals and in old animals. And it does so in the dentate gyrus, but not in CA3. So the dentate gyrus, which is critical for age-related memory loss, is affected by osteocalcin. Osteocalcin cannot mediate memory loss when function of RBAB48 is inhibited in the dentate gyrus. So osteocalcin produces its action, at least in part, through RBAB48. The behavioral effects of osteocalcin varies as a function of age. So aged wild-type mice show a decline in memory performance. If you now give an aged mouse osteocalcin, it enhances the memory performance. This not only holds for elderly mice, it also holds for young. So if you give a young mouse osteocalcin, its memory performance also gets better. Osteocalcin directly injected into the dentate gyrus increases the level of pKa, increases the level of phospho-Krebs and RBAB48. So the whole sequence of steps that leads to the activation of RBAB48 is activated. So it's a critical component of the switch that converts short-term to long-term memory. By contrast, knockout of osteocalcin mice have low amount of phospho-Krebs and low amounts of RBAB48. And systemic osteocalcin, if you inject it through the tail, gets through the blood-brain barrier, and you see accumulation of it in the dentate gyrus and in CA3. So we can recognize an antibody that reacts with both endogenous and recombinant osteocalcin that is present in the brain and clearly identifiable. We recently, in collaboration with Jared Kosenki, identified a receptor in hippocampal membranes, a G protein coupled receptor, GPR158, is a receptor that binds osteocalcin. It seems to be the receptor that recognizes osteocalcin in the brain. Knockdown of this receptor leads to reduction in the level of RBAB48 protein. And knockdown of osteocalcin receptor causes a memory deficit, and this memory deficit cannot be rescued by the osteocalcin. You don't have the receptor, you can't rescue the memory deficit. Over lifetime, the circulating level of osteocalcin decreases, but every time you exercise, you increase the level of osteocalcin. So bone is an endocrine organ. It releases osteocalcin. Exercising bone causes a release of osteocalcin and is absolutely essential for the aging brain. So if you swim every day, which I do, it's very nice, but it doesn't do a goddamn thing in terms of giving you this effect. This effect requires walking, running, jogging, doing something that really puts stress onto your bones. So osteocalcin injected in an old mouse scrambles around like mad, just loves activity. If you just inject saline, sleeps like most of us do when we don't have osteocalcin. So let me summarize. Osteocalcin released by bone ameliorates age-related memory loss through CREB1 and RBAB48. Because aging is associated with a decrease in bone mass, this decrease in osteocalcin could contribute to age-related memory loss. Conversely, this might explain the beneficial effect on cognition in aging of various exercises which build bone mass. The versatility of age-related memory loss strengthens the idea that age-related memory loss and Alzheimer's disease are distinct diseases. A sound body helps to assure a sound mind. So let me summarize. Age-related memory loss begins earlier than Alzheimer's disease. It begins in the dentate gyrus. Alzheimer's begins in the entorhinal cortex and then spreads. And the molecular defect decreases RBA-B48 and cyclic AMP signaling. No or minor loss of nerve cells with age-related memory loss. In here you have the classical A-beta toxicity, protein folding disorder, and a major loss of nerve cells. Treatment for age-related memory loss is to prevent the onset or progression of the disease. Physical exercise, walking, running, that increases the level of osteocalcin. A good diet, low in animal fat, control of things that that reduce your life expectancy, like high blood pressure and diabetes. Cognitive challenges. Come to grand rounds at 8 o'clock in the morning. Social involvement. That's absolutely key. So continue to work and to learn as physician scientists and enjoy your lives. Thank you very much. I will be glad to answer, try to answer any questions you have. Busby, I'm going to keep quiet, okay? Yes.
It's good to see you again. You were my professor in 2000 at Columbia.
And you survived?
You're better than ever. I had a question as you were talking about this, thinking about memory as it relates to acquisition and consolidation. The allocation is also CREB mediated, if I remember correctly. And I was wondering about how allocation, namely where memories go when we store them and how that's designated by the brain. How is that linked with aging, or have you looked at that?
No one has looked at that. It looks very likely, since explicit memory is sort of selectively affected, that this involves access to the hippocampus, which is critical for converting short-term to long-term memory for explicit memory. So some of the pathways involving the hippocampus are likely to be involved. People are starting to look at this. Now, this is a very important issue. Yeah. See, age-related memory loss, per se, is a relatively new phenomenon. People didn't think of it. When they saw age defects, they thought this was early stage of Alzheimer's. Now we realize it's a completely different entity and requires different approaches.
Yeah, it begs the question if it's getting less efficient as well as also just less, that you're losing that ability to push it forward.
Well, those are interrelated. Yeah, yeah.
So I'm gonna trail around behind you and pass off the microphone so that folks.
Of course, absolutely.
You're up.
So is the mechanism the same for male and female? One of your slides mentioned testosterone, but I know that in menopause there's some memory effects in the decrease in estrogen. So is there
You're absolutely right. Both of them are important. You're absolutely right.
So does the osteocalcin phenomenon?
Applies to women as well as men.
Okay.
Yes.
Aspon had a question, even though you told him he wasn't allowed to. Here you go.
I'm leaving.
I guess it's a rhetorical question, but presumably AP48 is reduced in the Alzheimer mouse because it's old, but if you upregulate it, does it do anything
No.
with memory?
No,
Nothing.
no. Alzheimer's is so overwhelming, it drives it down.
Thank you for that wonderful talk. Early on, you talked about some of the similarities between implicit and explicit memory on a molecular basis. And to me, that feels like it kind of roughly maps onto conscious and unconscious processing that's of so much interest to us as psychiatrists. And I was wondering if you could talk a little bit about the differences between implicit and explicit memory and how that relates to conscious and unconscious processing.
Well, implicit, explicit memory are examples of conscious and unconscious processes. Implicit often is unconscious, and explicit has a conscious component to it. But there are many other aspects of unconscious phenomenon that don't necessarily relate to learning, that relate to instinctual behavior and things like this. The Freudian unconscious is much broader than the kind of unconscious process I've been describing here. But I think it's important to begin to understand that, and people are beginning to study that. For example, people have done studies on, if I show you an image and show it to you only transiently, your occipital cortex lights up, but you're not consciously aware of it. But if you're allowed to see that image for a longer period of time, so you become consciously aware of it, you see that the information now propagates from the occipital cortex to other areas of the brain. So one of the things that happens when information moves from unconscious processing to conscious processes is recruits additional areas in the brain for further processing. That's really quite fascinating. And a whole group of people studying this now.
Yeah. Thank you for
I'll be back, okay?
a lovely and inspiring talk. Thank you for being here. Another recent phenomenon is space travel. And so I'm curious about whether or not osteocalcin has been studied in astronauts.
In what?
Astronauts.
Welcome to San Francisco.
No! I completely agree with you. This is coming along just now, so I don't think the astronauts are quite ready for this yet, but this will come.
Yeah, thanks for the great talk. I was wondering if there's been any research into like an osteocalcin agonist, osteocalcin receptor agonist that could cross the blood-brain barrier.
I'm sorry?
I wonder if there's been any research into osteocalcin receptor agonists that could cross the blood-brain barrier, and
Osteocalcin crosses the blood-brain barrier.
Oh.
So I think what people are working on, Gerard Kasenki was I've been collaborating with him, but he's the person who's been really developing this, is trying to develop a form that you can take orally. But that would be very nice to have. Yes, that's actually essential to carry this forward. Yes.
All right, we're moving down. Victor.
Yes.
One of the qualities of benign senescent forgetfulness seems to be that it's not an absolute deficit, but a delay in retrieval, so that
it has
we try to pull it up and it doesn't, and then all of a sudden, an hour later, a day later, it pops into consciousness. And I wonder if there's, first of all, any way to model that in the mouse, and two, what is that process about, where you're trying to engage in conscious retrieval, you can't do it, and then unconscious retrieval
brings it a little bit forward.
Spontaneously at some delayed point in time.
Yeah, there's no reason why you can't do that in the mouse. Then you can see where you give a stimulus that would normally elicit the memory, it doesn't do it, but if you do this 2 or 3 times, you can begin to see the memory emerge, and then you can actually see where was it hidden, what areas did carry it, even though they didn't express it, to the point where it conveyed it to the other areas that were necessary for expression. So those experiments could be done. They haven't been done. I just should point out to you one of the reasons many of the experiments on age-related memory loss, that experiments that come to your mind haven't been done yet, is this is a relatively new phenomenon. One used to think this as being, Alzheimer's disease. Now we realize it's really a much different and more benign form.
Thank you for your talk. I have 2 questions. One, more of a comment, but a number of years ago, some colleagues and I looked at changes in bone mineral density.
And bone density?
Bone mineral density, and showed that there was a nice relationship with cognitive decline, which was probably age-related.
Beautiful.
Exactly. So we at the time thought a lot of this was related to estrogen and testosterone changes, but
it's also related to bone as an endocrine organ.
Exactly.
Absolutely.
And the origins of bone have the same as in nervous system, of course. So it makes sense that there'll be a lot of common.
Right. But it's such a profound point that you make, because it emphasizes what is really good for you. I swim most days. I find this psychotherapeutic. But it doesn't do a damn thing for this phenomenon. You have to walk or run or something like that.
Weight bearing. Right. Weight bearing.
Weight bearing, not just weight bearing. Lifting weights wouldn't do it for you. I really think you have to move your legs.
My second quick question was: have you studied this in any other organ systems, such as non-human primates? Because this whole issue of that most animals don't get Alzheimer's is really a profound one, and yet we keep studying Alzheimer's in animals who don't get it because we manipulate with Alzheimer's disease, mice and rats, etc. So I'm curious whether you've studied this phenomenon in other organ systems.
I don't study primates, but I'm sure people are beginning to look at this in primates as well.
So while I walk down, I'm going to take chairman's prerogative and ask you a question. How did you get to osteocalcin? That once you knew RPAB48, how? I mean, it would seem like you could think of many different molecules that might.
Gerard Kasenki is in a building across from me. He was working on osteocalcin. We exchanged ideas and data, and we started to collaborate. This is how collaboration started.
There you go, collaboration.
Yes,
you hear that,
yes.
everyone?
By the way, even though I'm incompetent, I am trained as a psychiatrist, so if you want to ask me questions about professional paths, career tracks, and things like this, I mean, you may disagree with me, but I'd be glad to answer any, questions. I'm sorry.
Hi, I wanted to thank you for a really wonderful and extremely entertaining talk. It was wonderful.
You should come to all my talks. Sit right there.
I don't think I've laughed so much or so hard ever during a grand round in my entire life. So I'm an adolescent child psychiatrist interested in depression, and it's really, really one of those things that we don't have very many good treatments for. So I'm intrigued by all this work with osteocalcin and so forth, and I was wondering what your thoughts are perhaps about possible new antidepressants, and maybe our adolescents should be doing, should be running on a treadmill and having psychotherapy at the same time or something. But I'd be interested in your thoughts.
I think this is going to sound very superficial because I don't think about this very deeply, but I would think what we need in psychiatry is better measurements of process. So if you were to develop imaging methodologies that would allow you to identify the areas that are defective in their functioning, then you could see whether certain kinds of therapeutic intervention begin to affect. You might be able to see anatomical changes before you see behavioral changes, but that would encourage you to continue to do those changes because they're moving you in the right direction.
That's exactly what it is.
So I think I really think that this is essential, that imaging needs to be used more extensively and it needs to improve. The resolution imaging is not what we need to have ultimately, but this is a place that could do it. You've got terrific facilities here. Your Department of Neurology is also very distinguished. In fact, it's a very, very distinguished department. This place is unique in having a Department of Psychiatry headed by a major, major scientist, and having a Department of Neurology that is, as strong as the Department of Psychiatry, historically even stronger. It's a wonderful Department of Psychiatry. And then you got basic scientists like him around.
We better make sure Matt exercises so we can keep him around. No, the question is, there should be some epidemiological data available in the paralyzed individuals, especially when most of them start off young, since they can't exercise except for their upper body. Is there any evidence or studies done that shows that they have a greater cognitive decline?
That's considering it came from you, it's a very good question.
Unbelievable.
The answer for that should be obtainable. I don't know what it is. Why don't you look it up?
Hi.
Who's the person who has it? Yes.
I was hoping you could comment a little further on nutrition. You mentioned that low in animal fats. My sort of crude understanding was that there's been some reconsideration around that in recent years. I was hoping about the recommendation for diets low in animal fats. I was hoping you could comment a little further on nutrition and age-related memory loss.
I'm not sure that I'm really qualified to do that.
That's
I'm
fine.
sorry.
I'm a neuroscience PhD student here. It's nice to meet you. I was wondering do you think that mitigating some changes in levels of RBAP48 earlier on could kind of have a protective effect in delaying the onset of memory loss or even
I certainly think it's a very good idea. No one has really tried this, but I think there is no reason to believe you can't get a protective edge by doing appropriate things that increase the level of RBAP48 and things like that. No, that's very good. The other thing is that it's unlikely that you go through life not doing any exercise, and at age 82 you realize, I'm older than that, but you realize that you should be doing so and so and so and so. Most people who really lead an athletic life have been doing this from their youth on. Yeah, and that's very important for people to learn, to really continue to do what you're doing now as you age. It doesn't have to be walking, it can be, many people run, enter marathons, although that's an extreme which is unnecessary. Beneficial, but unnecessary. Yeah.
So we got someone right behind you.
Hi. I was very intrigued by your slide about the London taxi drivers.
Yes.
And I was curious if there's an understanding of, is that decrease in the hippocampus size once they retire related to the fact that they're not utilizing that specific set of knowledge of the geography of London, or is that more just related to overall decrease in interaction or?
Well, that's all part of it. And in their case, I think the evidence is pretty good that this is due to the fact that they're not using the spatial map function of the brain very much.
Hi, thank you. You mentioned the benefits of psychotherapy, and I wonder if we can consider as memory is an individualized expression of a shared representation, symbolic representation or experience. Could we consider that these physiological mechanisms are just a level of expression, but they don't represent the totality of cognition?
I'm sorry, I didn't quite understand.
That,
Did you?
sorry.
Can you try it again?
Sure. As memory is a complex phenomena, memory
Maybe is what?
a complex phenomena,
Yes.
and it's an individualized expression.
And what?
An individualized expression. It's the expression of.
It's individualized.
Yeah.
Yes. It's complex and individual, yes.
Yeah. So can we consider that there is something more than the physiological mechanisms?
No.
Why
Great
not?
question, and this is a great person to answer that
Yeah.
question.
It's all biological.
Why?
Every aspect of your mental process is biological. Everything Freud said is biological. And Freud said it's in the brain.
And don't you consider it's an assumption?
It's not an assumption, it's a fact. Biological and psychological are synonymous. Psychological is a reflection of a biological process.
And can it be the opposite?
No.
Everything is possible as an assumption.
The causative mechanism is the biology.
Why? I feel that it's
That's the
an
way.
assumption
That's the way.
It's the way we interpret the study.
That's the way we know the brain works.
Yes.
Beyond an interpretation. Okay.
Question here? Right here, behind you? Right here.
Yes.
Yes, so I was wondering if you could comment whether the process happening in the entorhinal cortex and the dentate gyrus, are they important for formation of the memories, or are they also important for the actual storage? Because it seems like when you remove the...
You make a very, very good point. This is important for the early transition from short-term to long-term memory. It is not the ultimate repository for long-term memory. That's probably in different areas of the cerebral cortex.
So we don't know where that is?
Well, in some cases we do have some idea. Prefrontal cortex is involved, yes. People are looking at that right now. That's a very important question.
Okay.
Yes.
We're going to take a long osteocalcin promoting stroll. Back over to you.
I'm glad you're giving me a workout.
We're helping you out. The internet tells us you can buy a bottle of osteocalcin for $815. And so we're kind of wondering, could you speak to the difference between perhaps, say it's a good quality pharmaceutical grade osteocalcin, the difference between taking a tablet versus all the other processes that are activated during, a high impact run or something like that, that may promote osteocalcin, but has a million other things
well,
that
we
make
don't
it
know
integrate.
that until we can compare the two, right?
Do you have thoughts offhand of
The
whether
only
that would
reason I
work?
think that the pill would do it is if you give osteocalcin to an animal, it does pretty well with it, okay? So it gets through the blood-brain barrier, blah, blah, blah. So and if a pill that is safe,
But you're injecting in the tail and things like that, right?
with that, that's to get into the vein, okay? in general, if it works that way, pills should work. Yes, but one could be wrong.
There's going to be a run on today. 500 orders from UCSF.
Hi, thanks so much for being here. I think that there's a lot of interesting evidence on the role of sleep in short-term memory and differential roles of sleep on implicit and explicit memory. So I was wondering if you knew or if you could tell us a bit about the role of sleep in memory over age.
Sleep and memory.
Yeah.
You know, last night I slept very little because I was with my daughter until very late, and then I woke up because I'm a New York time and God knows what. Fortunately, this was a ridiculously early hour for grand rounds, so I was prepared for it.
Thanks, Sarah.
But sleep is important, but it varies tremendously among different people. My friend Charlie Rose sleeps 4 hours at night, but he takes naps throughout the day. I also take naps. Before the opera, for example, my wife and I usually take a nap, 15 minutes, 20 minutes, and it's amazing what that does, you know, to really pep you up. I'm thinking of doing that more frequently, you know, like now.
Hi there. Going on with the idea that perhaps memory is, this is the gate for memory that you've found, right? The dentate gyrus people, and this is one target for one ligand. Are there other targets for memory
there
formation?
must be.
Exactly, and how are you going to get at them, and what are they going to be?
At the moment, I'm working with what I have, but there is no reason one can't. Usually, one doesn't, you know, just do a blind switch. One has a clue from some other kind of experiments that lead you in this direction. But you're absolutely right. You know, our understanding of memory storage has made a significant amount of progress, but it's got a long way to go. Yeah.
Hi.
Particularly when you consider something like the osteocalcin story, which has emerged in the last few years, and yet it's significant.
So your path towards the next ligand?
I'm sorry?
So then what would your path towards the next ligand be? Would it be imaging of aging, of the early onset aging brain?
Well, it's hard to look for ligand when you don't know what to look for.
That's exactly what I mean.
Yes.
So then,
No, I think what you have to, you can either take other things that are available that might, you know, look to you like they could have that effect, or just study what you have better and then get clues from this. The other thing you could do is maybe, since we have the receptor, to see does the receptor recognize other molecules as well. So is there a convergence in a common receptor?
We see, right here.
Right here.
Oh, I'm sorry.
Hi.
Hi.
We also see profound cognitive problems in people with schizophrenia in a decline-related fashion, and I'm wondering if there's anyone studied connections between this age-relating decline in memory and the problems in executive functioning or other processes that come with psychotic illness.
The cognitive function with schizophrenia disorder is not primarily a memory loss, although that's a part of it. So I don't think that is really exactly what one is looking at with age-related memory loss. It's not like people with schizophrenia just have an early onset of age-related memory. It's a different process. And it's more a distortion of perception than a failure to hold on to it. Yeah. Yes.
Hi, thank you so much for being here. You've been such an inspiration for all of us. So I wanted to ask you kind of a career question. What made you think back then to go from psychiatry to the lab? What inspired you to do that?
My wife. You know, I went to medical school with the idea of becoming a psychoanalyst. I got to know, I had a very good friend in college, Anna Chris, later Anna Chris Wolf, and her father, Ernst Chris, was a very powerful, well-known Viennese psychoanalyst. I was Viennese, he was Viennese, Anna was Viennese. So when Anna and I would read German poets to each other, we would occasionally think of a father, and I got to know him, and he inspired me about psychoanalysis. And I went to medical school with the idea of becoming a psychoanalyst. And in my senior year of medical school, I decided that even the psychoanalyst should know something about the brain. So I took an elective with Harry Grunfest at Columbia. At that time, practically no one was working on the brain. Many medical schools did not have a neurobiologist in the faculty. And I never had an experience like this before. I never worked in a lab. You know, I read about it in textbook, but it's boring. To actually do experiments, to use your own hands to set up an experiment, to see results, to talk about this, to gossip in the lab. I never had an experience like this. And Denise and I just decided to get married, and I was very upset because I said, you know, I could consider doing this for the rest of my life, but it's ridiculous. You don't have any money, I don't have any money, we want to get married. You know, I have to have an income, so I have to go into private practice. She said, money is of no significance. In the subsequent 61 years of our marriage, she's never repeated that once, but but that night, it had a major impact on me. Yeah.
I can't think of a better note to end.
I'm actually very inspired to be here because I think this department has the capability to really produce a significant change in the psychiatric picture. I think you're going to train a new generation of psychiatrists that are much more research-oriented, much more focused, and I think other departments will be influenced by this. Psychiatry has not moved very much in my career. I finished my training at the Mass Mental Health Center, which was then the highest of the high in 1965. Drugs were just beginning to come available. They haven't changed all that much. The field needs a push, and I think it's going to come from a place like this. Thank you very much.